to teach you. In this lesson, I'm going to teach you about enzyme structure and function. As you can see here, this is an enzyme, and enzymes are specialized proteins. You can see that this enzyme is currently in tertiary structure with alpha helices and beta sheets evident. Just like proteins, the structure of the enzyme is absolutely important for its function. If an enzyme is not properly shaped, it cannot properly function. More on that in a moment. Before we can start talking about enzymes, we need to talk about chemical reactions. Chemical reactions occur when two molecules interact and then change as a result. There's two components to a chemical reaction. First, we have our reactants, which are elements or compounds that enter into the chemical reaction. In this example, we have carbon dioxide and water. Those are our reactants. Carbon dioxide and water are going to enter this reaction, and they're going to form a product. The product in this case is an orange, and the product is carbonic acid. When CO2 and H2O react, they form the product carbonic acid. When we talk about chemical reactions in the body, we refer to the chemical reactions as metabolism. Biochemical processes that happen in our cell, bio meaning life, chemical referring to chemical reactions, and then the processes are just, of course, the reactions itself. We refer to them, again, as metabolism. Metabolism includes lots of different types of reactions. For example, dehydration synthesis, which occurs when monomers are put together to form polymers. We also have hydrolysis. Hydrolysis means that polymers are broken down into their constituent monomers. For example, proteins being broken down that no longer are needed, they're broken down into amino acids. Another example of metabolism or a metabolic reaction is cell respiration. Cell respiration occurs when we break down our food to create energy, also known as ATP. And a final example of a metabolic process or metabolic reaction is photosynthesis. Plants use photosynthesis to turn sunlight into energy in the form of glucose. In order to get a chemical reaction started in the body, or really anywhere, a thing called activation energy is required. This is a good thing because activation energy is required, chemical reactions can't just pop off whenever they like to. In your body controls these chemical reactions because activation energy is needed. And the activation energy is only lowered far enough for these chemical reactions to advance when enzymes are present in the cell. Again, activation energy is the energy required to start a chemical reaction. And another way to say this is that it is the energy required to get the ball rolling. As you can see here, the man at A is trying to push the ball up over the hill. That hill is what we call the activation energy. An example of a reaction that occurs in the body is an endergonic reaction. An endergonic reaction occurs when energy is absorbed into the process. This means that at the end of the reaction, there's going to be more energy in the products than in the reactants. So here you can see our products are higher up in the energy level, which means that energy was absorbed into the reaction. There's more energy in the products than there are in the reactants. Another type of reaction that's like this is called an endothermic reaction. So endothermic reactions are actually cold reactions. They feel cold because energy was actually absorbed into the reaction. An example of an energy releasing reaction is called an exergonic reaction. Exer means outside, so energy is released outside of the reaction. And in this case, when this happens, that means there's going to be less energy in the products than in the initial reactants. So you can see that when the reaction progresses, once you get up over the activation energy hill, when you end up here at the end, there's less energy in the products. Energy was released from the reaction. So as you can see, whether or not it's an endergonic reaction or an exergonic reaction, there's still going to be energy required to get the chemical reaction started. And again, this is a good thing because then energy or reactions aren't just popping off in the body. So how do we lower this activation energy far enough so that chemical reactions can occur in the body? We use biological catalysts known as enzymes. A biological catalyst is a substance that speeds up a chemical reaction in living things. Catalysts speed up chemical reactions by lowering that activation energy, so lowering the hill you have to push the ball up over in order to start a reaction. In this picture here, you can see the effect of an enzyme on a reaction. The line in red shows the amount of activation energy required without an enzyme present in the cell, and the blue line shows the energy required with an enzyme in the cell. So you can see that the activation energy with an enzyme is much lower right here. It's much lower than it is with a reaction that's occurring without an enzyme. The activation energy is much higher here. So because that activation energy is so high, without an enzyme in the cell, those reactions are not likely to occur because you're not likely to build up enough energy to get over that hill. Like I said before, enzymes are specialized proteins. So just like proteins, the enzyme shape is extremely important for its function. The thing that enters a chemical reaction with an enzyme is called a substrate. So we learned that reactants go into chemical reactions, but if an enzyme is involved in the chemical reaction, we call the reactants a substrate. Again, a substrate is a reactant of an enzyme catalyzed reaction. So an enzyme involved, then we have a substrate. On the enzyme, there's a special location called the active site, and you can see I'm highlighting the active site currently in red. The active site is the area where the substrate is going to bind the enzyme. As you can see, there's this very specific shape. The shape of the substrate must complement or complete the active site of the enzyme in order for the chemical reaction to occur. We call this the lock and key model. The key is the substrate because it enters the enzyme, which is like the lock. And once the substrate enters the enzyme, it unlocks the potential of the enzyme to create a chemical reaction. We call this the induced fit model because what happens once the substrate binds into that active site of the enzyme is going to snuggle very tightly around the substrate to keep that substrate in there through the course of the chemical reaction. Enzymes are very specific. We call this concept enzyme specificity. The substrate shape determines its ability to bind an enzyme. So for example, down here we have 4 different substrates. Substrate A, substrate B, substrate C, and substrate D. Only one of those substrates actually has the proper shape to fit into the active site of enzyme E. So only substrate C will work with this active site of enzyme E. Substrate D, substrate B, and substrate A all do not have the proper shape to fit into the active site of that enzyme. So if enzyme E is present in the cell, a chemical reaction including only substrate C will take place. This is great for the body because if certain enzymes are present in the cell, then certain chemical reactions can take place. But if we don't need a particular chemical reaction to take place, then the enzymes won't be present in the cell. For example, if your cell needs to break down fuel for energy, the enzymes that need to break down your food sources will be present. But if you have no food in your body and your body's not going to be breaking down food for energy, then those enzymes are not present. They're actually gone. They're broken down. They're turned off. So it doesn't waste any energy in the body. Here's an example of an actual situation of enzymatic control in your body. When you eat glucose, which is how we get our source of energy, that level of glucose in your blood increases as you eat. An enzyme called glycogen synthase enzyme is then turned on as the glucose concentration rises, and that glycogen synthase is going to turn the extra glucose into glycogen in your liver. So when you have too much glucose in your body turns on glycogen synthase, the enzyme, which causes your glucose to be stored as glycogen. When you aren't taking any new glucose in, so between meals, your blood glucose level is going to decrease and you're going to start to run out of energy for your cells. Well, that energy that you stored earlier can now be used by taking the glycogen out of your liver, breaking off those glucose monomers like we talked about before, and then releasing the glucose into your bloodstream to be broken down for energy. Glycogen phosphorylase is the enzyme that's going to be activated now. glycogen phosphorylase is going to break apart glycogen at the bonds between the glucose, and it's going to release glucose into your bloodstream. Glycogen synthase and glycogen phosphorylase are both enzymes that regulate blood glucose levels. Glycogen synthase is going to decrease the glucose level in your blood by turning your glucose into glycogen, and glycogen phosphorylase, the enzyme, is going to increase the glucose concentration in your blood by breaking apart glycogen when you need glucose for energy. So as you can see here in this diagram again, glycogen synthase is going to be responsible for turning glucose monomers into the polysaccharide glycogen. So you can see that happening on the right. And then over here on the left, you can see that when your body needs glucose, it needs to take that glucose out of the glycogen. So glycogen phosphorylase is going to come on in and it's going to break off those glucose monomers, increasing the glucose available for breakdown for energy in your bloodstream. So here's a basic overview of a metabolic pathway, meaning an enzyme pathway. So here you have your substrate binding to the active site of the enzyme. And once the substrate binds to the active site, we call this an enzyme substrate complex. So here, this green substrate bound to the enzyme, that's an enzyme substrate complex. This allows for the reaction to occur. Enzymes lower activation energy because they provide a place for substrates to come in and for a reaction to occur. So they decrease the amount of energy necessary for those molecules to actually break down. So once the enzyme has broken down the substrate, now we have two products still attached to the enzyme. So the enzyme is going to release the products back out into the cell, and the enzyme is going to make it from start to finish totally unchanged. This is awesome for the body because it actually helps us save some energy. Because every time a chemical reaction occurs, we don't have to remake the enzymes that were just used. They can be reused over and over and over again until they're no longer needed. So again, there's the enzyme substrate complex that occurs whenever the substrate binds to the enzyme. The enzyme is going to speed up the digestion or the breakdown of the molecule. In this case, it's a dimer because it's made up of two parts. And that's it. What is the biological significance of enzymes? Why are they important for the body? Well, here's some enzymes that are really important. Pepsin is the enzyme in your stomach that's going to break down food. So it catalyzes the breakdown of food. It speeds up the breakdown of food. Lactase in your body is what breaks down lactose sugar. For example, people who do not have the lactase enzyme in their body, we call them lactose intolerant. They can't have lactose because they don't have the enzyme, or the enzyme that they have is ineffective and cannot break down lactose. That's why they have to take supplements like Lactaid, because Lactaid is actually just lactase enzyme. So once they take that lactase, they have the enzyme in their body and it can break down lactose. So they can eat ice cream or drink milk or whatever. Another type of enzyme is called protease. Protease is going to break down the peptide bonds between amino acids in a protein, and it's going to allow for those amino acids to be recycled once a protein is no longer needed. For example, when you no longer need an enzyme in your system, protease is going to come in and break apart that enzyme and break it down so it doesn't have to do its job anymore. The last type of enzyme that's an example here of biological significance is an enzyme called synthase. There's all kinds of different synthase enzymes, for example, glycogen synthase. Synthase enzymes are just going to be enzymes that build up molecules or make molecules. So whenever you see the word synthesis, synthesis means to make. Synthase enzyme is going to make things. I hope that was helpful.